Hello, girls. Yesterday I shared with you one recording for the question number 1 to 10. Today we'll continue from question number 11. Before I start from question 11, I would like to remind you that when you're watching this video, you may pause at places. If you don't understand, go back and listen again. And if you are watching a slide with questions, so you should pause the video and try to answer the questions. That would help you to revise for your exam. So I will go straight away to the question number 11. We have finished everything. So we've done till here yesterday in the first recording. Question number 11. Now, question number 11, it is talking about surface area to volume ratio. So you need to know what is the effect of surface area to volume ratio, and how does it help in the functioning of the cell, and also it could be some calculations. So you should know both the things. This is based on figure 1 on page 224. 24. This is the figure 1. So first, what is a math connection? You know what? There are two terms which have been used here. One is the surface area and the other is the volume. You in math you've already studied what is the surface area and volume. So when we are talking about a cell, we take a cube. So the surface area of a cube is 6 times the area of its sides. So if the side measurement is 1 micrometer, because it's a cube, all the sides are going to be equal. So 1 micrometer times 1 micrometer into the number of sides. So there are 6 sides. So we have the formula. We use this formula and we get the answer as 6 micrometer square. This is the surface area. Now, what is the volume? Volume means a cubed. That means you cube the side. So in this case, the value of the side is 1 micrometer. So 1 times 1 times 1 will be 1 micrometer cube. So you should know how to calculate both these things. So if you have, supposing, if the cube cell grows to 2 micrometer, what would happen? So if it would be 2 micrometer, then we would have a surface area of 24. How did we get 24 micrometer square? Because 6 times 2 times 2. 2 times 2 is 4 and 6 times 4 is 24. So you have 24 micrometer square. What is the volume? Volume will be 2 times 2 times 2, just like we did here. The length into width into height. So cube has all equal sides, so 2 multiplied by 2 multiplied by 2. That will give you 8 cubic meters cube micrometer cube, sorry. So the surface area is 24 and the volume is 8. Now what is the ratio of surface area to volume? They mentioned surface area before, so you write 24 ratio 8. What is the common factor here? You can reduce it to the simplest form, and that would be 3 ratio 1, because you'll divide it by 8 on both sides, so it will be 3 ratio 1. This is less than it was when the cell was small. When the cell was smaller, when it has side 1 micrometer, the ratio came to 6 ratio 1. When the side is 2 micrometer, the ratio is 3 ratio 1. That means it has become smaller. It is less than this. If the cell continue, if it will go on growing, the ratio will keep on becoming smaller. It will decrease. So, as the cell grows, its volume increases much more rapidly than the surface area. So, it is why this is not very effective, because it is difficulty in supplying nutrients and expelling enough waste. So, if the size is more, the volume increases. It's difficult to supply nutrients. Surface area does not increase with that rate. So small cells, now what is the advantage of small cells? Small cells have a higher ratio of surface area to volume and can sustain themselves more easily. So if a cell gets too large, transport of nutrients and waste by diffusion will slow down. Therefore, cells divide before they become too large. So this is one way to keep them small. So cells, they don't grow in size much. They will divide, and they are like small cells. Here, this is the effect of surface area and volume on the function of the cell. Cells exchange materials through their surface area. Nutrients, oxygen, what are the materials that they exchange? They take nutrients, oxygen, water that enter the cell. Waste products go out of the cell, and cell membrane represents the surface area of the cell. So, exchange is taking place through the surface area. Volume of the cell determines its needs. Volume represents the amount of material inside the cell. As volume will become more, cell will need more nutrients and produce more waste. So surface area does not increase as quickly as volume, as I told you. When a cell grows larger, its volume increases faster than its surface area. This causes the surface area to volume ratio to decrease because the volume is increasing at a faster rate than the surface area. So the ratio will become less. Now, what is the effect of it on cell functioning? So, this ratio of surface area ratio volume, it allows materials to enter and leave the cell quickly. A low surface area and volume ratio slows down the movement of substances. So, large cells may not get enough nutrients or they may not be able to remove the waste efficiently. That's why it is not good, and how the cell like works so that it can work efficiently is that it divides and forms new cells. Okay, so next is now the same thing, explain the effect area of volume ratio to the function of the cell. So I already told you why cells remain small. Cells have a larger surface area volume ratio. This enables efficient diffusion and transport of substance. A larger surface area volume ratio will make the diffusion more efficient, and the transport of substances will be more efficient. So, cells often divide when they become too large. What are the adaptations to increase surface area? Some cells have folds. How do they increase the surface area? How do the cells? So some of them have folds. They're folded. The cell membrane is folded. Some have projections, that means from outside. It is like growth outside. Or they have microvilli, small finger like processes on the cell membrane. These all increase the surface area. These adaptations improve the exchange of material without increasing volume. So, the larger the surface area to volume ratio, the more efficient the cell is at exchanging substances. As it will grow, the ratio will decrease, making transport less efficient, and this limits cell size, and it because of this it triggers cell division. So cell division happens. So here this is a small cell, the nucleus, and as it will grow, if you find the surface area to volume ratio, you will see that it keeps on decreasing. You may be asked to calculate, like they may give you a question where they have asked what is the ratio, surface area and volume ratio of a cell with side this. So you'll have to calculate it and then choose the option. Here are some questions. As I told you, you will pause the video and you're going to read the questions and try to see the answer. Try to check whether you know how you got this answer. What is the ratio of surface area to volume of the cubic cell below? Now, here they've given you a cell and they've asked you the ratio. So, you will need to do the calculation. Here it is 2. So, surface area will be 2 times 2 times 6, and then volume will be 2 times 2 times 2, and then you will find the ratio. Write it in its simplest form. Okay, here it is 4, then the ratio is what is the ratio of the volume is 3. So you'll have to calculate. Definitely, there would be one question like that. Which cell has the lowest ratio of surface area? As the size will increase, the ratio will become less, smaller, decrease. So the third one, which is the largest in size, will have the lowest ratio. Okay, then Carlos is studying human skin cells under the microscope during science class. He asked his teacher why cells are small. So the response would be what you can understand, that larger cells cannot efficiently transport nutrients. So these questions you will do. Okay. That was question number 11. Now we come to question number 12. Question number 12 is going to talk to you about the cell cycle. You know that the cell cycle has three main stages. This is figure 4 of page 226. This is figure 4. So there are three main stages in a cell cycle. First is interphase, then mitosis and cytokinesis. This is the whole red color. This one is the lighter color. This is interphase, M is mitosis, and C is cytokinesis. So you can see the major part of the cell cycle is interphase. And interphase is again, you can see, divided into three substages: G1, which is called gap 1, S synthesis, G2 gap 2, and then mitosis and cytokine. In gap 1, nothing is happening, no division is taking place, only the cell is growing and it is performing normal function. So growth is taking place in G1. After G1 in S, now here something happens. In synthesis, the DNA gets replicated. Replicated means it is copied. It is copied and it forms copies of its own type. So DNA gets replicated in S. Then for G2, the cell is now preparing for mitosis, for dividing. It prepares for mitosis. So in interphase, first the cell growth, then the synthesis, that is replication of DNA, and then the preparation for mitosis. So this is the first figure. Okay. Then here you see cell reproduced by cycle growing by a cycle of growing and dividing. This is called the cell cycle. Each time cell goes through one complete cell cycle, it becomes two cells. It divides. After one cycle, it will divide into two. Now, what is the duration of the cell cycle? Cell cycle might complete in as few as 8 minutes, while in some it takes a long time, maybe up to 1 year. It takes place about 12 to 14 hours for most normal actively dividing animal cells. It can be 12 to 14 hours. So there are three main stages, as I told you. Interphase, cell grows, develops into a mature functioning cell, duplicates the DNA in its nucleus, and prepares for the division in interphase, in the S. And interphase, as I told you, divided into three stages: gap 1, gap 2, and synthesis. Gap 1, immediately after the cell cycle, growing, carrying out normal functions, and preparing to replicate DNA. Synthesis, when cell copies its DNA. Replicate means copying. Then in gap 2, when the cell prepares for the division of its nucleus, the cell takes inventory and makes sure that it is ready. It will check everything done, DNA copying done, and everything ready, then it will go for mitosis. Then what happens in mitosis? This phase here, M. It is a stage when the cell cycle in which the cell's nucleus and nuclear material divide. DNA is already divided. Now the nucleus is going to divide. So here the nucleus will divide and the nuclear material is going to divide. So the replicated genetic material will separate and the cell prepares to split into two cells. This is mitosis. In mitosis, only two cells will be formed. So the key activity of mitosis is accurate separation of cells. They enable DNA. This enables the cell's genetic information to pass into. So all the genetic information is passing on to both the cells that are formed. We call them the daughter cells. They are genetically identical cells. So what does mitosis do? It increases the number of cells. Organisms also use mitosis to replace damaged cells, to repair damaged cells, to replace, remove the damaged cells, replace them. Also mitosis is used. Okay. So sometimes when our hand or gets cut, then the body starts healing it. New cells are formed, and the existing cells, they just form under the scab. The repair work goes on, and then the injury is healed. And this also takes place, mitosis, for maintaining chromosome number remains the same, and when organisms, they undergo asexual reproduction. So this is your question number 12, and here are some questions based on question number 12. Which stage in the picture below represents the preparation of mitosis? So mitosis, I told you, G2. So all this, please go through these questions. All this is there. So stop your video, read the questions and see the answer. Then go on, start watching again. Now we come to question number 13. 13 is again talking about mitosis and cytokinesis. So, what are the events that are occurring within each stage of mitosis? So, we know that mitosis has been divided in two different stages. You have PMAT, I told you, PMAT, prophase, metaphase, anaphase, and telophase. These are the stages of mitosis. So you should know what is happening in each stage. So this is figure 6 on page number 228. If you can't see, watch it clearly here. You can go to your book and have a look at it on page number 228. So first phase, this is the interphase. Then here comes the mitosis. So after interphase, the mitosis will start, and that is prophase. First is prophase. This is prophase. Remember to identify the picture, because they give you the picture and they say which stage is it. So try to remember how the figure looks like, and then you can, like, identify it. So this is prophase. The material which is chromatin material here, the chromatin, it has condensed and formed chromosomes. You see the nucleolus has disappeared and the chromosomes are formed. So nuclear membrane disintegrates. There is no nuclear membrane now. Nucleolus has gone. Nucleolus is not available. Chromosomes, they condense. Condense means they become thick, and spindle apparatus begins to form. You can see here the spindle apparatus starts forming. This is prophase. Second is metaphase. In metaphase, meta, you can remember middle. M, meta, M, middle. So all the chromosomes are lined up, aligned in the middle of the cell, that is at the equator of the cell. They are aligned at the equator, chromosomes. So if you see this figure, you know it is metaphase. When you see this figure, you know it is prophase. Now what's happening in metaphase? In metaphase, the chromosomes, they line up along the middle of the cell, and spindle fibers, you can see these fibers, they attach the centromere of chromosomes. They attach to the centromere. This is the centromere here at the poles. So the chromosomes are attached to the centromeres. Then we come to the next stage, that is anaphase. Now see the chromosomes which are like X shaped, they are separated and they're making two V shapes, and they're going to the two pole side. So this is anaphase. In anaphase, these are the chromatids, the chromatids, they are pulled apart, and these chromatids, the tubules, the spindle fibers, they shorten, and when they short and they become short, so they pull the chromatids separate, they separate the chromatids, and they are pulled to the opposite poles of the cell. So the centromere, is that part where these are joined together. So and the centromere is attached to the spindle fibers, they are shortened, so they pull and they separate. So this is anaphase. Then comes the telophase. Now the chromosomes have separated, the chromatids have separated, they reach to the other poles. Now what happened? They need to change into two parts, divide into two parts. So chromatids reach the opposite ends of the pole in telophase here. New nuclear membrane. Now the nucleus is formed again. Here it had disintegrated, here it forms again. New nuclear membrane forms around each set of chromosomes. So around each set we get a nuclear membrane, and the chromosomes again start uncoiling. They begin to uncoil. So this is the telophase. Prophase, metaphase, anaphase, and telophase. After the nucleus has divided, formed two nucleus, then comes cytokinesis. You can remember the cytoplasm divides. So here the cytoplasm divides, the cell will split into two genetically identical daughter cells. So the cytokinesis is different in plant cells and animal cells. This is very important. In plant cells, the cell plate forms, dividing the daughter cell. So in plant cell, a plate will form in the center and then it will divide the cell. But in animal cells, a furrow, cleavage furrow, this like this. A cleavage furrow is formed and at equator of the cell, and it pinches inward and the cell divides into two. So you should know the differences between the cytokinesis in plant cell and cytokinesis in animal cell. So these figures very important. Prophase, nuclear membrane disintegrates, nucleolus disappears, chromosomes condense, and spindle apparatus begins to form between the poles. I just explained you the metaphase. This is the anaphase. I just explained you all this. And then this is the telophase. Chromosomes reach phase and the nucleus divides into two. Nuclear membrane forms and nucleolus also appears. The same thing again. These are the same description again. I think I've already covered it. Have a look at all this. Okay, so these are the questions based on this. Pause the video and watch and read the questions and check whether you understand and you know the answers. See here, this is, they will see which below illustrates the stages of mitosis, which of the following represents a telophase. Now you know which one is telophase here, where the nucleus is forming. So you need to know this one is which one, metaphase, this is which one, anaphase, and here this is the prophase. What are the structures labeled here? What are these called? These are the spindle fibers. What stage of mitosis is shown in the drawing above? In the drawing above, it means this one, I think. Or this one here. They're talking about this one. It is metaphase. Describe the action of the cell chromosomes during metaphase. What's happening? They line up in the middle. Which phase of mitosis are sister chromatids pulled to opposite cells? Here they pull to opposite ends. This is anaphase. Which of the following is part of the spindle apparatus? The ester fibers. These are the ester fibers, like a star. These are the ester fibers. This is again the same thing. You should, not the same question, but you can watch the video, okay? Okay, now we come to, it says specific events that occur within each stage of mitosis and cytokinesis. We already studied mitosis. Now this is cytokinesis. Here, you know, in cytokinesis, plant cell, animal cells different. I just told you here is a plant cell, a cell plate is formed. And in animal cell, a furrow is formed. So that is how cytoplasm divides to form two daughter cells. We already did this. Now we come to again the cytokinesis. Towards the end of the mitosis, cell begins cytokinesis. Cell cytoplasm divides, resulting in two cells with identical nuclei. So this is again the same figure, plant cell, cell plate and furrow. Again the same cell plate, and here is the cell plate formed and furrow. Okay, so next is questions based on this. So cytokine, how is it different in plant cell, animal cell? They generally ask you. These questions are taken from the previous exams. Like here, what is Which of the following is occurring during cytokinesis? Now, cell